I'm really impressed what I've seen and heard today. It's really exciting, the vibrancy, the energy, the enthusiasm, and I think, you know, people like Nick and Ian and many others, and more recently Peter and Nari, need to be really congratulated. And what's happening out of the Auckland Regional Council, you know, it's pretty aspirational. And if you can keep carrying this forward, you've got a snowball that's rolling, and you'll be able to do things in time. So I think I needed to say that. And I worked and have worked with many people, and I've benefited from others working with people, and many students and many colleagues, some are with me here today. So a lot of this is not my own work. And Peter sort of indicated this earlier. It's really important to understand these organisms, their background, where they're from, what they're doing. We need to understand them fully. And it took over 40 years for us to determine what was killing Jarrah. 40 years. And we still might be missing things in that system. Recent work that Trina's been doing is showing that there are phytophoras out there that we're only finding now as a result of next generation sequencing. And we can't culture them, for example. Maybe they're playing a bigger role than what we anticipate. The other thing with Jarrah, it's actually quite resistant. And it really requires some wet feet to drive the process. So predisposition in Jarrah is very important. And whilst this was happening in Jarrah, we had our eyes off the ball. And we were losing many other hundreds of species, actually thousands of species in the southwest of Western Australia, which were really, really susceptible in our natural ecosystems. And so this is something you also need to be aware of. What's happening to other species as you're looking at PTA or the organisms involved with the Kauri dieback. And just a few pictures. Peter showed some of these. This is the Stirling Range National Park, and you can see nice healthy bushland. There's 1,500 species, endemic species here. What after bushwalkers have carried it up, all these plants die. It changes structure, function, everything, and it moves down slope in these grey areas. Out areas are all the species that have been killed. So we just left rushes of sedges, and all the flowering species have disappeared. So the impacts on fauna are substantial. There's another example in another woodland on our south coast, 2,000 odd plant species, and about 41% of them are highly infested. And the important ones, the flowering ones, the ones that provide a lot of ecosystem function and services. This is just around Perth. We've got healthy Banksia woodland, and as the pathogen goes through, it changes. You can see the changes. It's substantial. And for 40 years, this was happening in front of people's eyes whilst they were looking at Jarrah. And then suddenly people started to realize, wow, what's happening elsewhere? The Jarrah forest, Peter's shown you that, healthy Jarrah forest. The declining Jarrah forest is substantial. This is something that you might be left with. These trees grow 600 years as well. Chewett, Peter showed you some of this again. Another Phytophthora species, Phytophthora multivora, which you have here. You have here too. And we named it appropriately, multivora. Its host range is expanding all the time. And I feel quite strongly, and we're seeing you isolating it here, that it probably is playing a role in your Kauri as well. Don't just put all your emphasis on PTA. They may be working in concert together, synergistically. Peter alluded to this as well. And I also mentioned the Jarrah at the beginning. You know, it needed wet feet actually to kill Jarrah. There was predisposing factors there. So this decline spiral is very important. And we had some very good questions from the audience. What other things have happened? You know, climate change. In Western Australia, we're 20% drier in some areas. We're about just under a degree hotter in the last 25 years. This is something that must certainly be playing a role in when we're starting to see trees die. So there are a lot of predisposing, inciting, and contributing factors we need to understand, especially with trees that live 1,000 years, 1,700 years we heard from Bruce Burns this morning. You know, they're long-lived, they can't move. So actually teasing out these sort of questions are very important. So this is Kauri, your Kauri. I feel passionate about your Kauri too, and walking around. You know, you want to hug them. You know, the healthy ones. It's sad to see them dying. So how does that decline spiral I showed in the previous slide actually work here? You know, there's more going on than just the pathogen, and it's important. So it's really nice seeing Bruce and Utgard working in this space. They're important people to support. They're asking the right questions. So what I'm really enjoying seeing here is the multidisciplinary approach coming into the research towards PTA. I think this is what's really nice. You're starting to cover lots of different areas, and you're starting to ask the sociological questions as well, and it's about the people. And this is what you're here for today. And we need to be much more cleverer at asking the right questions, and I think I'll cover some of this. So, as I mentioned earlier, don't forget Multivora, Cinnamomi, and the other Phytophoras that are out there. You know, having been around the world and seeing what we're seeing in Western Australia, these other Phytophoras are there, they're doing something. And I just put those two rings in. You know, there's other plants dying here. You know, are they hosts or are they being killed by a Phytophthora? We need to understand what's going on with other plant species. I've put this in, Multivora. It kills a wide range of hosts, and by a sudden collapse, many different species, and big trees as well as small trees. And yes, I've asked that question. The other thing we're starting to see is we see in another Corymbia, another eucalypt relative, another major pathogen, which causes cankers. It's a basidiomycete. But we're finding very close association with Phytophoras in the root system. And this canker, this pathogen here is endemic. It's been around. We think it's endemic. It's been around with Mary forever, but now it's starting to kill trees. And we think these Phytophthora species are actually driving the process. They're weakening the roots, less vigor, less nutrients. They're taking out the mycorrhizal fungi, and the trees are suppressed, and they're getting killed. So there are other associations we need to look at. I put this up. This is Reddy Trina's work. And many new Phytophthora species. The ones in orange are 11 we've described in about the last 4 or 5 years. And the green ones are being described as we speak. And the interesting thing is that all of them associated with dead and dying vegetation. We don't know if they're endemic. We don't know if they're introduced. We'll be finding this out as we get more isolates. And I know that you're looking, and Nari and Peter will be doing this, and you'll be having sort of similar scenarios. So what are we doing now? We actually spend a lot of time mapping Phytophthora in our landscape, and you talked about this. And often it's difficult in our landscape to interpret everything. So we have, you know, dieback free, uninterpretable as a result of fire, for example, and red is infested. We're lucky we base this on susceptible species, so we can put people on the ground and they map the presence of Phytophthora based on susceptible indicator species. You haven't yet found these. And then where they actually blaze trees on 3 sides, and on the side that isn't blazed is dieback free. So any people operating on the land, forestry activities, mining activities, etc., and no, don't cross the line without clean down. So we come up with fairly comprehensive maps. This looks pretty awful, but in and around these red dots, there are large areas that are still uninfested. The other thing I just wanted to say is we're discovering now with Cinnamomi many asymptomatic hosts, herbaceous annuals and perennials, which are really good carriers. They're actually propagating those resting propagules that we heard Stan and Peter talk about. They're actually producing huge amounts of inoculum, but they don't show symptoms. And maybe this is something you could think about with, you don't have as many species as us, to actually look at these understory species, herbs, etc., and isolate from them. They might actually be your indicator plants. You might be able to use them to map where your agathis, taxon agathis is. Even though they're not showing symptoms, you might be able to come up with a strategy to sample certain numbers to actually develop a mapping system. And this to us was a major finding, especially when we're talking about eradication and managing the pathogen in different systems. We've heard about phosphite, and Ian's made a really compelling story about phosphite. It works very, very effectively in many plant species around the world, especially in our system. We apply it from the air, we apply it by a backpack, by injection, and also as a paint. It works. We apply it in disease-free areas adjacent to areas that are heavily infested. So we tend to treat adjacent disease-free areas with it rather than the infested, or a little bit of a buffer of infested, but not the whole infested area, because often it's too late. But it does stop this rate of spread. It doesn't kill the pathogen, it really reduces the rate of spread, but saves many, many of our plant species. And in the process, in areas where we've got only a few left, we're able to go in and collect seed and of rare endangered species or cuttings, and we can transfer them and put them into areas which are disease-free if we're going to lose them. So, which is something I think we need to be doing. So, what about the community? What about people like yourselves? And there's been a lot of activity with local friends groups, community groups, and little friends groups just protecting small areas of native bushland, for example, in the city or in peri-urban lands, and also in our rural environments. And a few years ago, there was about 2005, there was a big project called Project Dieback, which was all about stakeholder engagement, all the people operating on the land. How do we manage this pathogen? And I want to talk about this a little bit now. We know that Phytophthora cinnamomi is spread across about 1 million hectares in 5 natural resource management regions. And all the areas in red are the infested areas. And we get worried about showing maps, actually, of where it's present, because the pathogen moves quite quickly. And we need to remap it every 2-3 years if we want to be sure where the pathogen is present. And providing people with maps makes them feel safe. Well, it's here, it's not there, but in actual fact it could have moved in a short period of time. So what we've done is, because we know we've got limited resources, we're starting to prioritize areas. And we're now looking at the protectables, and there are many areas across those 5 management areas which people want to protect, but we're not going to be able to protect them all. So we're looking at selecting just 100 sites across the whole area, based on size, based on what plant communities are there, and how many are rare endangered species, for example, or priority threatened species. Where they're located, if close to urban centres, how many roads run through them, are people using four-wheel drive motorcyclists, bushwalkers, those sorts of things. So here's some examples. An area with a little bit of Phytophthora in it, obviously highly protectable as a biodiversity hotspot here, another area through here, but also in highly infested areas. They've selected sites based on community engagement and stakeholder discussions. These are small areas amongst the infested that they believe will be protectable in the next 100 years, based on, key threatened plant species or fauna. And so a lot of different metrics go into selecting these sites. And then we think we might be able to get resources from the government and other agencies put into these areas to stop the spread of the pathogen. And also in some of these areas, we can then prioritise. If we see another red spot in here, which we've seen recently, we go in, spot infestation, and I heard that from Ian, I think. We go in and we try and eradicate it. We nuke the areas to stop it spreading so we find a small spot infestation of, say, 1/2 hectare. We will knock out 2 hectares, destroy everything. But that saved it moving into 10,000 hectares. And so this mapping and prioritization is actually quite important, because then they can mobilize the resources. So once we've picked a priority area, there's a lot of mapping on the ground. They will rip up tracks or trails and bulldoze the tracks so other forward drivers can't use it. They'll change the direction of the roads if they need to. So they'll put down wash-down stations into certain areas to also help stop the spread. In each of these areas, a lot of surveys, understanding the different stakeholders. This is just one example with the SES, the State Emergency Services, and it's just one of the many questions. And, for example, don't really understand what good dieback hygiene is and how can I apply it to my organisation. Hygiene takes too much time, and the costs can't be justified. Don't have anywhere to wash down vehicles and machinery. Machinery hygiene is not effective in fighting the spread of dieback. So the biggest constraint those stakeholders said was, we don't have anywhere to wash down our machines. So we've gone through and asked lots of those sorts of questions, and they inform the managers what to do next, where to put the resources. And that feedback is often very important. and I've died. I didn't touch anything. So, I can go now if you want me to. But in actual fact, like you, we do a lot of signage as well, and consistent signage. When we started, we had those 5 resource management regions, and they all started having different signage. And the community, the wider users, the tourists, everyone, were getting really confused by different signage. So we've worked really hard at consistent signage across all stakeholders. He's a good communicator, but he can also drive these machines. He can't do without you, Nick, in the Phytophthora world. Should we just live with it? Go there. Okay, so signage is really, really important, and also information is why we should be, why we want you to wash down, for example, why we want you to wash your clean boots, to show some of the key assets you will be protecting by cleaning down and listening to the signage, and they show the loss of species, they show diagrams of what Phytophthora is doing to the landscape. So there are consistent messages. The signage also says you're driving out of the park now, if it's a muddy road, the nearest washdown station is X or Y, so you're encouraging the people to go to a washdown facility to get their vehicle washed before they go into another national park or another area where Phytophthora is known, where it's disease-free, for example. So the other one is a lot of communication packages, and you're doing that very well. Out there, there's a stand with a lot of very useful information, relevant information that is usable, and you can put to different stakeholders. So stakeholders like plantation growers. This is blue gum plantation, but their land is often in priority areas, and they have to move through native bush to get to their plantations, and under the FSC, the Forest Accreditation certificates, they have to justify they're doing something sustainably. So we work with them to implement Phytophthora hygiene management. They're a different type of stakeholder with different demands, so we work with them to ensure that they're not spreading the pathogen. So every stakeholder is different. Gravel for building roads. We now know areas that are disease-free, so we make sure roads are built with disease-free gravel, but we wouldn't access that gravel during wet periods, only dry periods. So those sorts of messages. I've talked about this sort of signage, I'll move on. Consistent messaging I've talked about, but quite clearly here, where you can wash your vehicle. It's important, and luckily you're doing that. You haven't had that constraint, but we had that constraint early on. And the different resource management groups, theirs was the best signage. No, ours is the best signage. They really didn't want to come together early on to implement something that was consistent across all of the different shires or land care groups. So just more of the same. And again, this one's just indicating some of the things you'll be maintaining if you do implement good hygiene. So you're doing all of this, handouts, information, it's all very important. Stickers you put on your vehicle, keeping the message out there. The other thing we put a lot of effort into is trying to come up with messages every 1 month that gets picked up by the media. People, if they don't hear about it after 6 months, oh, we thought we had cured that. And no. So in actual having a communication strategy where you could be hitting the media some way, we find quite useful, and quite a lot of effort's being put towards just keeping it in people's faces with good news as well as the bad news. Good news messages are very important, and I feel you've got a lot of good news messages that could be being sold out there. School children really important. They're our future, and they will actually tell their mums and dads to clean their boots when they walk. Dad, you can't do that. You know, so you communicate with them. They actually work very efficiently.
and engage. So we actually have education packages with primary schools that are taken up, and it's taught in their curriculum as well, especially in areas where the schools are adjacent to native woodland or in areas where Phytophthora is a major problem. This is a new thing that's come on board called DIDMS, the Dieback Information Delivery Management System, that you can actually access. Are you laughing because we call it DIDMS? Yes. Yeah, it's a bit like a drop bear. So it gives important message, persistent messaging. It shows where Phytophthora is and it isn't. It informs people where they can go to access information, interested community. It's something that's consistent, and that's really important. And the messages are the same. The messages are the same and consistent. And so, really, there are a lot of things we need to be doing. Detailed mapping is really important. Know where the pathogen and where it's not. And I think it's very important to know what other pathogens, what other Phytophthora you've got as well. Not just put the emphasis, because they suddenly might become major problems. Sometimes they're what we call sleeper pathogens. They just need to develop, get enough inoculum, settle in, if you like, and then boom! They have a heyday. So knowing what you've got is really important. Know your hosts, symptomatic, asymptomatic hosts. Implement some of those forest quarantine areas, and you're doing that. You're talking about shutting out areas. And we're doing that with our top 100. I think we'll be going down that route. And we did in the early days, but it'll be happening again. The hygiene and management activities, you're doing that. In some ways, you're better than us. Phosphide application, I think don't underestimate it. I mean, yes, you've got to get the cultural and other social things under the board, but you know, you've got a 1,000-year-old tree with a lesion up here. I think you can save that tree based on what we're doing in Western Australia, if you treat it. Eradication, we do use eradication where we need to. And I was quite interested in hearing the previous but one speaker that you are looking at eradication. We can do it successfully. We can get rid of the pathogen using different management techniques. Communication and training with the stakeholders is really important. We do a lot of that on days like this, but in smaller groups. And we also mentor and train friends groups as they come into being. We help them, we assist them in the process. And doing what you're doing today, engaging the wider community. Thank you.